Biology students, this is 11.1, Complex Inheritance and Human Heredity, which we touched on the end of the last chapter when we were discussing speciation and polyploidy. So there's 3 sections here: basic patterns of human inheritance, complex patterns of human inheritance, and chromosomes and human heredity. The big idea for the chapter: human inheritance does not always follow Mendel's laws. The main ideas of each section. Section 1, the inheritance of a trait over several generations can be shown in a pedigree. Section 2, complex inheritance of traits does not follow inheritance patterns described by Mendel. And Section 3, chromosomes can be studied using different chromotypes or using karyotypes. I knew I'd get it. As always, we have our essential questions to the chapter, as well as a review and new vocabulary. So let's dive in with section 1 and start talking about recessive genetic disorders. A recessive trait, if you will remember, is expressed when the individual is homozygous recessive for the trait. Homozygous means the same, right? So if we go back to our Punnett square from before, that means it has a capital or a lowercase. In both those situations, that's a homozygous trait. But this is asking which one is the homozygous recessive. That's right, the lowercase. So those with at least one dominant allele will not be able to express the recessive. And individuals who are heterozygous for a recessive disorder is called a carrier. That means they're showing the dominant trait, but they're carrying inside of their genetic code the recessive trait. So they may not be able to see it, but they can certainly pass it on to future generations. Cystic fibrosis is a disorder that affects the mucus-producing glands, digestive enzymes, and sweat glands. Chloride ions are not properly transported out of cells of a person with cystic fibrosis, and it causes mucus excretion that clogs up ducts in the pancreas, interrupts digestion, and can block respiratory pathways in lungs. Another genetic disorder that is recessive is albinism. It's caused by altered genes resulting in the absence of skin pigment, melanin, in hair and eyes. Individuals with albinism have very pale skin, white hair, and pink irises. Tay-Sachs disease is caused by the absence of the enzymes responsible for breaking down fatty acids called gangliosides. Gangliosides accumulate in the brain, inflating brain nerve cells and causing mental deterioration. Galactosemia is also a recessive genetic disorder, and it's characterized by the inability of the body to digest galactose. Basically, inability to digest milk products. So if you're lactose intolerant, that's a recessive gene. Somebody gave it to you. A dominant genetic disorder is Huntington's disease. It affects the nervous system, causing gradual loss of brain function. It occurs in 1 out of every 10,000 people in the United States. Achondroplasia causes small body size and limbs that are comparatively short. It's also caused by an abnormal gene that can affect bone growth. In your online textbook, there's several interactive tables. Make sure you're using them and practicing them because this is all on the test. So a pedigree is a diagram that traces the inheritance of a particular trait through several generations. So if you've ever mapped out your family tree, this is the family tree of genetics. And as you can see, it has a legend just like other types of maps. And although they're pretty much basic across the board, sometimes they are different. So make sure you're always referring to the key. So in this one, a normal female is round and yellow, and a normal male is also yellow, but he's square. A female who expresses the trait being studied, meaning she has it, and everybody can see it. It's her phenotype as well as her genotype. She is still round, but she is maroon and the male is maroon as well. And if a female and a male don't express that gene, but they are carrier for it, then they're split half and half. And these are things that will not be on every single one of your legends, so you have to memorize them now. This means generation, parents, siblings. Roman numerals are your generations, and Arabic numerals are individuals in a certain generation. Pedigrees can be used to examine both recessive and dominant genetic disorders. Information about an individual's genotype can be inferred from the phenotype of his or her parents and offspring. So in analyzing pedigrees, inferring genotypes is knowing physical traits can determine what genes an individual is most likely to have. And predicting disorders is record keeping, helps scientists use pedigree analysis to study inheritance patterns, determine phenotypes, and ascertain genotypes as well.
A pedigree is like a family tree. It can show information about an inherited trait passed across generations. And this one is actually a small pedigree of us. Well, our human forms. See, that's me, my sister, my mom, my dad. I'm not arbitrarily picking random shapes to represent us either. In a pedigree, the circles represent females, squares represent males. One way you can remember that is the letter C for circle comes before the letter S for square in the alphabet. Alphabetically, the letter F for female comes before the letter M for male. I've got to think about letters of the alphabet, though. This trait being tracked is that it's recessive. Recall that in typical Mendelian inheritance, dominant alleles, if present, will lead to the expression of dominant traits. Recessive alleles only are expressed when the dominant allele is not present. Now, fact number 2 about this trait is that it is an autosomal recessive trait. Just a reminder that autosomal means a chromosome that is not a sex chromosome. In human body cells, there are 46 chromosomes. The first 44, 22 pairs, are autosomes. The last 2, 1 pair, are sex chromosomes. So this trait is not sex-linked since it is autosomal, and that means it does not need to be written as coefficients on the sex chromosomes. So what is the trait we're tracking? Attached earlobes. Yes, you might not realize it, but look around and you'll see that humans, they have free or attached earlobes. Although we want to point out that there may be more than just these 2 categories for earlobes. And while this example is used often in basic genetics, there's probably more to this than just one simple gene. For our example, let's assume a simple gene trait, and that free earlobes is dominant, meaning at least one dominant allele must be around. Attached earlobes is recessive, showing when no dominant allele is present. So if we were to put the genotypes next to each of these shapes, what would they be? Well, the shaded ones would be easy, because we just mentioned that attached earlobes is the trait we're tracking, and it's an autosomal recessive trait. So if we use the letter E, then these shaded shapes must be lowercase e, lowercase e. Any capital dominant letter, and the individual would have to have free earlobes and not be shaded. So let's look at individual 2 in the first generation. That's our father. He's not shaded, so he can't be little e, little e. What about big E, big E? Well, there's a problem. See his children? Us? Each child must get an allele from each parent. So if I received a little e from my mom, then I had to get the other little e from my dad. Therefore, he can't be big E, or he'd have no little e to give. His genotype must be the heterozygote genotype, big E, little e. He's what we call a carrier, though. He still has a phenotype of free earlobes because of that one capital, but he carries the lowercase allele. Now, that's just one tiny pedigree. Let's look at a big family reunion. Well, an imaginary one, because I have to confess I don't really know whether our relatives have free or attached earlobes. I thought about sending a survey to them, but it just felt a little awkward. So here we go. Big, giant, imaginary family of relatives. Okay, just to make sure you understand this, how many siblings does my dad have? Well, look, here's my dad in generation 2, number 4. He has 3 siblings, all brothers, right here. What is the phenotype of my paternal grandfather? Well, look, here's my dad, here's my dad's dad, That would be my paternal grandfather. And because his square is shaded, that means his phenotype is attached earlobes. Okay, so let's go ahead and label all these shaded shapes with the genotype e, since we know that's the trait we're tracking. Now take a look at generation 1, individual 1.
Now, when doing these on your own, I would highly suggest doing this and marking them with the alleles that they should have. Always use pencil because you might change your mind.
Would be my paternal grandmother. What's her genotype? We know it's not e, or her shape would be shaded. But if we went with big E, could that still work? Yes, all the offspring could get a big E from her and a little e from my grandfather. But what about big E, little e? Would that work too? Yes, because the children could still get a big E from her and a little e from my grandfather. It may be less of a probability, but it's possible, and therefore we must list both, that she is a big E, or big E, little e, because we don't know. All the offspring of my paternal grandparents, though, they're going to have to be big E, little e. Remember, they have to get an allele from each parent, and that means they're going to have to pick up that little e from my grandfather. They will be heterozygotes, and that's the only option here. Pause this video and try to solve the right side of this pedigree now. Ta-da! Imaginary family done. So how'd you do? Well, here are some of the tricky ones. Did you see that generation 1, individual 4, has to be a carrier only? Because if not, then the shaded individual children would not be able to get the little e that they have, because they have to get a little e from both parents. How about individual 9? This female married in, but that's not the reason that she can be either big E or big E, little e. If you look at the children, they aren't shaded. So while they will have to get a little e from number 8, as that's all number 8 can give, the other capital letter can be obtained from number 9 regardless of whether she's big E or big E, little e. Remember, one option may be more likely, but if it's possible, you need to include both. Now, remember how we made a big deal about how this was an autosomal pedigree? Well, what if you're dealing with a sex-linked trait and therefore a sex-linked pedigree?